Okay, take it away. Thanks, Paul.
Okay, thank you. So we'll finalize this session with talk by myself. So I'm Paul Parren, head of R&D and executive vice president at LAVA Therapeutics. And I'll talk about our bispecific gamma delta T-cell engager platform. In this case, having an antibody targeting PSMA for the treatment of prostate cancer. And this is the first time we're presenting these data. Maybe it's just more in general, why targeting V-gamma9 or why targeting gamma delta T cells? So, of course, we know that CD3-based T-cell engagers or bispecific antibodies that can engage T-cell generally on basis of their binding to CD3 for tumor cell targeting have been effective and they're also very widely investigated. And we heard some talks about that in this conference. But we've also seen that these types of engagers are related to serious side effects, including cytokine release syndrome and on-target off-tumor toxicities. So we think it's time to move forward and develop a next-generation T-cell engager platform that rather than activating all T cells, now selectively target a specific T cell subtype that has a more specific role in terms of having cytotoxicity against tumor cells. And we think a very interesting or particularly prominent T cell type that could be targeted this way are the V-gamma9 V delta 2 T cells. And why these cells? Well, the V-gamma9 V delta 2 T cells are a T cell type, gamma delta T cell type that plays a central role in antitumor immunity. It's a pretty prominently available type of T cells, so 1 to 5% of your peripheral blood T cells alone are gamma9 delta T cells. And this is a fairly homogeneous and highly cytotoxic effector T cell population. They have a monomorphic receptor, so they recognize their targets similarly, and I'll show you in the next slide how that goes. And they have a natural ability to recognize and kill tumor cells. So they are already set up for tumor preferential killing, and we can make use of that using our bispecific antibody platform. They also have an ability to present antigen to other cells and orchestrate downstream immune responses. So we think this type in particular is of high interest in terms of our ability to recruit it with a bispecific antibody for tumor cell therapy. On top of that, it was shown some years ago that V-gamma9 V delta 2 T cells are present in tumor tissue. And there's this study on the left there that was published by Gentles et al. Nature Medicine a number of years ago, where they surprisingly demonstrated that gamma delta T cells, and specifically, as later was shown by Tosolini, and that study is shown on the right, gamma9 delta 2 T cells are present in the tumor microenvironment of many different tumor types. And in essence, their presence thereby also correlates with favorable outcome. So Gentles, for example, showed on the left, that if one looks at the presence of leukocytes in different tumors, then gamma delta T cells most strongly is the one leukocyte subset that most strongly correlates with survival in patients. And on the right, then Tosolini showed indeed the presence or abundance of these cells in many different tumor types, including hematological as well as solid tumor indications. And I should mention their prostate cancer as the second one, where it demonstrates that there indeed is a high abundance of these gamma9 delta T cells, specifically in prostate cancer. Now, what we did at LAVA is develop a technology we call the Gammabody platform. So it basically is a bispecific antibody technology where one arm of the bispecific binds the delta 2 part of the V-gamma9 V delta 2 T cell receptor, and the other arm binds the tumor antigen. And in the studies I will present today, these are targeting either PSMA or EGF receptor. This cross-linking by of the gamma delta T cells to the tumor antigen gives a conditional activation of the T cells. So only upon cross-linking can we see activation, and then and this then leads to potent tumor cell killing. The Gammabody platform is fully modular. We can combine it basically with any existing antibody targeting a tumor antigen, and thereby we think we have a really very feasible way to develop novel antibodies targeting novel T-cell engagers that could lead to off-the-shelf therapeutics for streamlined development and protection. But the Gammabody platform is further of interest because, as we said, the initial step in activation is the induction of targeting and activation of gamma delta T cells to the tumor cells, which gives an immediate response as the cells degranulate and potently kill the tumor cells. But we also demonstrated that there's downstream effects, so the expansion of gamma delta T cells downstream, activation of additional immune cells, as classical T cells and NK cells, and as I mentioned, there is potential for antigen presentation to T cells and thereby the potential for more durable responses. Now that said, I'll now talk about our studies on PSMA. So we have selected PSMA as a cancer target for our first solid tumor program. Clearly, PSMA is a very well characterized and validated tumor antigen. We heard a lot about it already today, and there is significant overexpression of PSMA in nearly all primary and metastatic prostate cancer tissues. Also, prostate cancer remains a major area of unmet medical need. Once the tumor is metastasized, then the 1-year survival drops down to low levels, and clearly novel treatments are urgently needed. Now, prostate cancer is the solid tumor indication with the highest relative abundance of tumor-infiltrating gamma delta T cells, so therefore it seems also a good candidate for a first program with this novel technology. In gamma delta, the presence of gamma 9 delta T cells, as shown by Dosolini, as I mentioned earlier, in prostate cancer has shown a strong correlation with improved patient prognosis, and a graph from that paper is shown there on the right. And there are additional rationales for bispecific gamma delta T cell engager approaches, including the early clinical data from CD3-based programs, where initial clinical activity has been observed, but also cytokine release syndrome has been observed as a dose-limiting toxicity. I show you some data today where we demonstrate that our PSMA targeting gamma delta T cell engager give potent lysis of PSMA-positive tumor cells, whereas normal prostate cells are being spared. Also, I should mention that the gamma delta T cell population that we target does not contain any immunosuppressive T regulatory cells, so therefore these cannot also not be activated, and we therefore do not believe that the technology will lead to the suppressive effects that have been observed with some CD3-based engagers, in which activation of Tregs and dampening of responses has been apparent. Now, PSMA is overexpressed in prostate cancer, as I mentioned, and there's some studies we have done ourselves here where we see higher overexpression on the tumor. And also we have demonstrated that gamma 9 delta T cells are present in the tumor tissue and actually at similar levels as they are in normal tissue or in PBMC, and thereby further supporting the data that were previously presented by others. So both the target expression as well as the effector cells that we require are present in these tumors. Now, we developed a PSMA targeting Gammabody, and the structure is shown here on the left. It's called LAVA 1207. It's a bispecific antibody with an Fc domain and consisting of 2 domain antibodies. VHH is one targeting the delta 2 part of the gamma delta T cell receptor, and the other targeting PSMA. The Fc is silenced to prevent of non-specific T cell activation due to binding to Fc receptors on additional cells. On the right, we show that there's strong binding to gamma 9 delta T cells with an affinity of around 1 nanomolar, so there's high affinity binding, and the binding to the PSMA with this antibody is in the order of 20 nanomolars. This then leads to very strong picomolar cytotoxicity and degranulation induced by LAVA 1207. As shown on the left, we're looking at CD107 expression on gamma-9 delta 2 T cells in the presence of LNCAP tumor cells, and we see that degranulation occurs with an EC50 of around 16 picomolars. This also leads then to potent cytotoxicity on the right. Again, now lysis of PSMA-positive tumor cells with an EC50 of around 17 picomolar. So high potency in both activation as well as lysis of tumor cells. Now then we did some studies where we investigated the activity against both normal cells as well as tumor tissue. So here is one example with the predecessor of LAVA 1207, so the non-humanized antibody, domain antibody, bispecific domain antibody, where we see that on the left there is lysis of tumor cells expressing PSMA in the presence of V-gamma-9 delta T cells and the bispecific antibody, whereas on the right we show that normal tissue, normal prostate tissue is being spared. We also performed studies using autologous gamma-9 delta T cells. So these were studies done with samples from radical prostatectomies, where we then took the cells, made single cell suspensions, and then again combined the tumor cells as well as the normal cells with gamma-9 delta T cells in the presence of LAVA 1207 and looked at both degranulation and tumor cell kill. So on the left you can see that indeed the antibody induces strong degranulation of gamma-9 delta T cells, both obtained from the tumor environment as well as from PBMC, so autologous PBMC obtained from the same patient can be activated. And on the right, again, similarly to the previous study, we show that indeed there's lysis of the tumor tissue, whereas the normal tissue, again, was spared. So there's tumor preferential killing also by autologous gamma-9 delta T cells, either derived from the periphery or derived from the tumor microenvironment. Now, how does this work? So why is there tumor preferential killing? We think this is due to the expression of certain ligands for receptors that are involved in V-gamma-9 V delta T cell recognition of tumor samples. And these ligands are overexpressed in cancer tissue, as shown here. So on the left, we show that two receptors, DNAM1 and NK2GD, are expressed on PBMC, as well as normal gamma-9 delta T cells, as well as gamma-9 delta T cells obtained from tumor tissue. And these then can interact with receptors on the tumor cells. On the top, we show receptors for DNAM, Nectin-2, and the poliovirus receptor are overexpressed on the tumor. In the middle, we show that MIG-AAB, as well as the ULBPs are overexpressed on tumors. So these are ligands for NK2GD. And on the bottom, we also show that the BTN3A1, which is butyrophilin, so this is the natural ligand for the V-gamma-9 V delta T cell receptor, which on top of that needs activation by the binding of phosphoantigens, which are typically present at higher levels in tumor cells. So all these three signals, we think, contribute to the relative tumor specificity of V-gamma-9 delta T cells, and we retain this tumor specificity in the presence of our bispecific antibody. Now also NK2GA is overexpressed on gamma-9 delta T cells. Recently it was demonstrated that NKG2A expression was shown to identify a subset of V-gamma-9 V delta T cells that exert the highest antitumor effector function. So it's encouraging that indeed these cells are present at higher levels in prostate cancer tissue. On the right, we show these tumors also express enhanced levels of HLA-E, which may help the tumor escape may escape the killing by gamma-9 delta T cells, but we also know that once gamma-9 delta T cells become activated, that this signal can likely be overcome. We demonstrated in vivo efficacy of our antibody in a human prostate cancer xenograft model, MixXeno5, the MixXeno model shown here, where we both gave tumor cells that expressing PSMA, as well as gamma-9 delta T cells obtained from PBMC from a human donor. So the xenograft was mixed with the PBMC and then implanted in the mice, and then we performed treatment on 4 occasions, as shown here. In the middle, you can see that indeed only when we have PBMC present, as well as the antibody, we induce significant protection against tumor growth, and this also leads to increased survival, which is significant. So the antibody can protect against tumor growth in an in vivo model. Now, next we explored the non-clinical safety of this platform, because clearly what we set out to do was to make a molecule that has an increased safety profile, and thereby we would like to achieve a wider therapeutic window than has been observed with current CD3-based T-cell engagers. So to do this, we explored the safety using a fully cross-reactive surrogate Gammabody molecule, and we did this in various flavors. Initially we made Gammabody molecules that didn't have an Fc, that targeted CD1d, CD20, as well as EGFR. But after that, we also now explored more recently EGFR Gammabody that has an Fc domain, mimicking the structure that we also have in our PSMA Gammabody. We didn't do the study with the PSMA Gammabody itself. First of all, our antibody doesn't cross-react, but also PSMA is fairly poorly modeled in macaques with no expression in prostate, so we didn't think that is a relevant model to look at. EGFR, which is a abundantly expressed antigen, as you know, in relevant tissues, is more relevant from this perspective. And also a CD3-based T-cell engager targeting EGF receptor, which is a BiTE generated by Amgen, has been shown previously to give very severe toxicity in non-human primates, and it was shown to be lethal within 56 hours of dosing at only doses of 31 microgram per kilogram and above. So we think we thought that indeed an EGFR Gammabody, in this case also containing an Fc domain, was the best way to study the potential safety of this technology in non-human primates. So this was the surrogate molecule that's what generated. It's a fully cross-reactive molecule that consists of a single chain Fv targeting V gamma-9, which is cross-reactive between human and monkey gamma-9 delta T cells, and also it contains a fully cross-reactive domain antibody against EGF receptor, and apart from that, the structure is similar to the PSMA-targeted Gammabody I've just described you before. On the right, we show that this molecule can induce degranulation of gamma-9 delta T cells, as well as cytotoxicity against EGFR positive cells. Target binding and in vitro activity, therefore, were confirmed. And this molecule is called LAVA 223x227, and just identifying the two different chains that were used, and the Fc domain is fully functional but silenced in terms of effector function. So the non-human primate study that was performed is shown here. We used 3 groups of monkeys, each consisting of 1 female cynomolgus monkey, and then we dosed this antibody at relatively high doses of 1, 5, and 23 milligram per kilogram. Of course, these are not any doses that would be relevant for these type of molecules for in human studies, but here we just wanted to push and see the full safety window of these types of molecules in a relevant model. The antibody is given by IV infusion, and then we dosed 4 times once weekly. Euthanasia and necropsy was done 2 days after the last dose. The PK analysis shown that we had sustained high levels of the cross-reactive Gammabody against EGF receptor, as shown also there on the right. Only for the third dose, at the low dose group, we saw some fast decay, suggesting that there was an ADA response in this group. No evidence for ADAs, antibody responses in the other groups were observed. The elimination half-life was between 84 and 127 hours, which is quite similar to the half-life of a canonical IgG against EGF receptor in this model. We saw systemic exposure that was dosed proportionally between 1 and 23 milligrams, and we did not see an accumulation of the drug after repeat dosing. Most importantly, we did not see any changes in general health parameters, clinical chemistry, hematology, or histopathology. So no toxicity at all, and this compares extremely favorably to the previous data with CD3-based agents against EGF receptor, which were, as I said, lethally toxic at microgram per kilogram doses. We saw accumulation of the antibody, which was dose-dependent in relevant tissues, as shown here. So we see increasing staining in the tissues shown here, going from 1 to 23 milligram per kilogram. And basically the last box here shows tissues where we did an additional incubation with exogenous antibody just to demonstrate the specific staining. So you see these staining patterns are almost identical. On the right, we showed the presence of induced cytokines in the plasma of the monkeys. These were observed at very low levels, mostly after first dose. We saw some IL-2, some IL-6, virtually no interferon gamma at any of the doses. The amounts observed were picograms per milligram, so these are many logs difference, many logs lower than what was previously demonstrated for CD3-based T-cell engagers targeting EGF receptors. So again, very benign profiles, and also no observation of any cytokine release syndrome in these monkeys. The flow cytometry data, we show pharmacological activity of the antibody. Both T cells, as Vy9 delta T cells, were reduced after each dosing, as is usually observed in these types of experiments. We did the CD3 positive T cells returned back in circulation at the same level. We see that there was some decrease in Vy9 V delta T cells, which we believe is due to extravasation of these antibodies as a result of their activation. And indeed, at the bottom, we demonstrate that the circulating Vy9 positive cells indeed show signs of activation, and therefore pharmacological activity, as shown by expression of activation markers CD25 and CD69. So then in conclusion, our study shows that a fully cross-reactive Gammabody targeting EGF receptor, so activating Gamma9 delta T cells in monkey with a surrogate Gammabody, was highly tolerable. Repeated dosing was done up to doses of 23 milligram per kilogram. We did not see any clinical signs or any other relevant safety-related effects. The induced cytokines were very low. And this was consistent with the absence of signs of CRS. We showed pharmacologic activity of the antibody. PK was expected, similar to canonical IgG1, and immunohistochemistry demonstrate accumulation of the antibody in relevant tissues. Just one final slide. We did do some pharmacokinetics of LAVA 1207 in cynomolgus monkey. We believe it's a suitable model. It interacts with cynomolgus FcRn, so we can look at half-life, but it, of course, does not bind cynomolgus PSMA or cynomolgus Vy9 V delta T cells. So it's a non-target binding study. What we observed was that the half-life of the antibody was between 6 and 7 days, which is fully in line with the expectation for non-target binding regular IgG1 antibody. And also this was also in line with data from a human FcRn transgenic mouse model. With that said, we're now in a first-in-human trial with LAVA 1207. It's a phase 1/2a open label study to evaluate the safety, tolerability, pharmacokinetics, and pharmacodynamics, as well as immunogenicity and tumor activity of this molecule in patients with refractory metastatic castrate-resistant prostate cancer. The design of the study is shown on the bottom. CTA and IND filings have been cleared, and currently the trial is open for recruitment. With that, I'd like to end. So just in summary, so we think we have strong scientific and clinical rationale for a tumor-targeted engagement of V-gamma9 Vδ2 T cells. We have a modular platform. We differentiate from a pan-T cell approach because we believe our system, our Gammabody platform, will have result in a much more benign toxicity profile, preferential killing of tumor cells and sparing of normal cells, and also we avoid the engagement of immunosuppressive cells. Overall, we believe we'll have a wider therapeutic window, which may lead to more optimal dosing. With that said, I'd like to end and acknowledge my colleagues at the Amsterdam UMC, where we have a research group, and specifically also at LAVA Therapeutics. I mentioned my colleagues here who have contributed to this work. So with that, I'd like to end and take any questions that you may have.
Can take your questions right now. I don't see any yet in the chat, but if anyone online wants to ask a question, we have time right now. And while we're waiting for questions, just remind everybody that we have buzz sessions in the foyer with breakfast tomorrow morning starting at 8, and we'll start here at 9 a.m. and we go till approximately 5 p.m. And so far there are no questions. Last call. Oh, great. Okay, can you step up to the microphone so people online can hear?
Thank you. Very interesting talk and very exciting to see the lack of toxicity with this strategy. Do you have any hypotheses on why, especially in your EGFR targeting model, the gamma deltas really don't seem to elicit that horrible response that alpha beta T cells can?
Yeah, we do. We really think this is a unique quality of these effector cells. So gamma-9 delta T cells, as I said, they are known as effector cells to protect us from infection, but also from cancer. And they have this immunosurveillance function. So also the natural ability is an ability to sense the difference between a tumor cell and a normal cell. So one of the ways this is done is by this recognition of butyrophilins, so the BTN 3A1 and 2A1 that are expressed, typically overexpressed on tumor cells. But these butyrophilins also need to be activated, and this is due to the presence of phosphoantigens that can accumulate in tumor cells because they are mevalonate pathway and their metabolism is dysregulated. So this combination gives you some tumor specificity. On top of that, there are some other molecules like NK2GD specifically, and also DNAM, as I demonstrated, that also are being used by these gamma delta T cells to sense the tumor cells. So we think that even in the presence of our bispecific T cell engager, because clearly this provides a very high affinity interaction and cross-linking, but even that signal in itself, although it can activate the gamma delta T cells, and we can see this activation, as we also see in the monkey, but a next step that may not then lead to a functional synapse. So we think really the combination of these co-stimulatory molecules together with the bispecific creates that in the end creates a functional synapse that leads to tumor cell lysis. So that's the hypothesis. We have now data that show this expression of these molecules. We're doing some more structural analysis to get the finger behind this. But I should say, it's really also the data that demonstrate that this is the case. We have, obtained data with many tumor samples for many different patients where we looked at the activity against tumor cells and normal cells, and this is a differentiation we see again and again.